Welcome everyone and Happy New Year. Happy to see many of you here today. This is the first session of the BioClock Academy lecture series in this new year, and we are looking forward for new exciting topic also for this season. My name is Juana Georgiana Roos-Oswald. I'm a postdoctoral researcher in the BioClock Consortium, and together with Hannah Brooks, which is a PhD student in the same consortium, we will host the session today. Just for the ones who joined it the first time, the BioClock Consortium is a Dutch-funded organization which aims to study the biological clock on 3 different levels: health, society, and environment. And the BioClock Academy is a lecture series organized within this consortium. And it is addressed to anyone which is generally interested in this field, but especially is made for early career researchers. And its aim is to introduce the basic concepts and equip you with the proper theoretical knowledge by experts in the field. The lecture series takes place every 3rd Wednesday of the month. And the time frame for today, we will have a 40 minutes talk, followed by 50 minutes of discussions. So roughly 1 hour. We will close at 5 p.m. And during the talk, you can write your questions in the chat so that we can discuss them at the end. And now I will give the floor to Hannah, which will introduce our speaker today.
Hello, everyone. So also a warm welcome from me. It's nice to see that you are all here today for the topic that we will discuss today. So the light entrainment of biological clocks. And this talk is given by Professor Roelof. For the people who do not know Professor Hut, he's an associate professor in chronobiology at the Faculty of Science and Engineering at the University of Groningen. And he has really been dedicating his scientific career to this field of chronobiology, mainly interested in the topics of neurobiological backgrounds of these rhythms, but also evolutionary and more ecological aspects of these rhythms. And he has performed his work mainly on mammals, but over the years has also been including insects and birds and humans. So a wide range of knowledge. And we can also see this wide field of interest within our BioClock consortium itself, where he is part of both our societal as our environmental cluster. So based on this wide range of knowledge, I think we will have a very nice session today. So let's just start. Roelof, the floor is yours.
Thank you. I hope you can all hear me. Last year I was promoted to full professor, so I'm very glad that happened. So, and yes, I am working on a lot of different topics, although today I will try to focus on not so much on clocks, but more on light and how to measure light. We thought it was a good idea. I evaluated also this with Luc Slangen recently, so I'll try to focus on that. So now I have to share my screen. Let me see, where did it go here? And I have, I think, 39 slides ready for you. So I hope you bear with me. Let me see, this should go. Maybe, where do I leave this? Somewhere at the bottom, maybe. So yeah, I like this introductory slide because it really sets the stage for biological clocks that of course synchronize to the light-dark cycle, which is induced by the rotation of the Earth. And of course, there's seasonal cycles as well, because the Earth rotates around the sun. So clocks and lights are very important. You have seen, by the previous presenters, you've seen this phase response curve. The circadian clock is synchronizing to the external light-dark cycle because it is sensitive to light when it's expecting darkness. So in the subjective night, which is a part of the phase of the biological clock, we see that the clock can shift if light falls on the... Do you see my pointer, by the way? Do you see my arrow?
Yeah. Yes.
Yeah, okay. So you can see that in the beginning of the subjective night, light will induce delays, and at the end of the subjective night, light will induce advances. And it's easy to see how this is synchronizing clocks. If your clock is slow with a period longer than 24 hours, then the light in the morning will synchronize it and adjust it to exactly 24 hours. And if you have a fast circadian clock that has a period shorter than 24 hours, light in the end of the day will cause delays that are sufficient to synchronize it exactly to 24 hours. And because of the slope of this phase response curve, these systems will stabilize when all is well. So that is how the circadian clock synchronizes. And we call this classical entrainment theory, or phasic entrainment, or entrainment by phase shifts. So light is important, environmental light is important, and I'm going to dedicate a relatively large number of slides on natural light, because we have a lot of people that work in nature, and then I'll continue later on with artificial light. So natural light, of course, comes from the sun, but also other elements. And here we have a little schedule that I made a long time ago. Here we have time of day, and we see that due to the Earth rotation, it seems like the sun is moving if you are viewing it from a point of view on Earth. And this is calculated for the Netherlands. And you see that the sun rises at 0 degrees above the horizon, and that's where the center of the solar disk is at the horizon. And then it reaches its maximum in midday, which due to our social time is not always 12 o'clock, but much later in the Netherlands. And then it sinks again and it sets. Now, this moment, the time points where it is at the horizon, 0 degrees, we call sunrise and sunset. But actually there are interesting other definitions that sort of could indicate lights on, if you want, in nature, and lights off. And those are civil twilight, nautical twilight, and astronomical twilight, each with 6 steps below the horizon. Now, why is that important? I'll show you in the next slide. Basically, what happens in this scheme is that if you change seasons, if you go into the winter, we can see basically that the horizon is lifted relative to the sinusoidal movement of the sun. And in summer, the horizon is sinking. And that causes, of course, the width, the hours of daylight to expand in summer and to decrease during winter. But also latitude is important. This seasonal change, the amplitude of the season, actually you could say the amplitude that the horizon moves is of course changing with latitude, causing, for instance, continuous darkness or continuous light if you are above the equatorial, above the polar, if you're in the polar regions. Now, here we have a sunrise. This is an old graph that Serge Daan made in 1975, where he measured the light intensity in lux with a lux meter, a simple light meter. And you see that the logarithmic light intensity increases during sunrise. And here are indicated these different twilights. And you can see that the light intensity is almost already at its maximum, maybe 1 log lower, 1 log unit lower than the maximum at sunrise or sunset. So if you were to define lights onset and lights offset in nature, it's much better to use civil twilight, because that is sort of halfway, close to halfway, that steep increase. So around civil twilight, that's where the light intensity changes most. And this is actually, yeah, so this is in degrees above the horizon, but you can of course set a time axis on that as well. So what we did, actually what I did in my PhD is try to measure the spectral composition of that light. At the time, it wasn't actually measured. It wasn't that clear at all how the spectral composition changes over the course of the day. So here we have time of day, and we see that at sunrise, at civil twilight at dawn and civil twilight at dusk, indicated with the arrows, you can see that different wavelengths are coming in. And you see the timing when these wavelengths are coming in differs a bit by wavelength. So the longer wavelengths in the visual range, they're up here, they all come in as more or less one band. So this 440, maybe 420 to 700, that's where the visual system of humans is sensitive. And you can see that all these radiations, all these photons in that wavelength and those colors come in relatively at the same time. And if you go into the UV light, then you see that the sunrise, the ultraviolet sunrise, if you want, is actually quite a bit later. And then the sun sets already quite a bit earlier. So this is something we need to explain. And we see that these relationships also on summer days are maintained. The other thing that's important, you see this activity bar, this is the activity of a diurnal animal. But of course, you have to realize that if animals are in burrows, that solar pattern can have very different effects. If you are in the open air, you'll have some light intensity even at night, whereas in a burrow, it can be more dark during the night. And then if you have, for instance, if you are burrowing during daytime, there are different patterns like C, D, E, and F. And so maybe a nocturnal animal can have a light pulse only in the evening when it is emerging, and maybe a little bit of a light pulse or no light pulse in the morning when it's going back to its burrow. So the light patterns that an animal can see is really depending on its behavior. The other thing that is important here is that the spectral, the ratios between different wavelengths may change over the course of the day. And I can actually show that much better in the next slide, because Manuel Spitjan is a person who knows a lot about these light spectra. And he has this publication 2016 that I advise you to maybe look up in Scientific Reports, where he analyzed different solar spectra relative to, so at night, and then relative to the position of the horizon, so astronomical twilight, nautical twilight, and civil twilight, and then during daytime again. So here we have a first view on how solar spectra would look like during daytime. We'll see a bit more about that later on. But we can also see that if you are moving, if you look at civil twilight or even nautical twilight, when the sun is 12 degrees below the horizon, you can see that there is a huge increment in short wavelengths. So we may think that the sunset is red, but in fact that is I would almost say an optical illusion, if you want, or at least a subjective illusion. I will explain that. So in fact, if you look at the spectral composition, you see that the whole dome of the sky is actually shining deeply blue, irradiating deep blue lights, even into the UV light, as you can see here. And here at nighttime, we have another phenomenon. We have a lot of infrared light, and then we have also some enrichment here in the UV light that we need to explain. We have an interesting peak here, which is, I think, if I recall well, the oxygen resonance peak, if you call it, excitation peak. All right. Now, that is in the rural areas in the US. It was measured in the rural area in the US with hardly any artificial light. If you look close to the city, I think it was Philadelphia, you see that there is a lot of changes in that spectrum, and you can see a lot of artificial light coming in. Of course, when the sun is out during daytime, the sun is the strongest light source. There's not much of an effect. But if you look here around nautical twilight, astronomical twilight, and especially at night, you see that there is a lot of visible light coming in, which is due to the artificial light in humans. And you can actually, looking at the different peaks, you can say something about what type of light source those are, and those are mainly street lights. Now, why is that twilight giving so much blue, so much short wavelength? In order to understand that very briefly, we have to understand two types of scattering. As actually one type of scattering is most important, that's Rayleigh scattering. So Rayleigh scattering in the atmosphere is caused by particles, and those particles are very effective to scatter short wavelength light. The shorter the wavelength, the easier light rays are scattered. So basically what's happening is that when the sun comes in the atmosphere, there is scattering of UV light and blue light, and then the longer wavelengths like green and red, they're penetrating much more easier. So they have sort of a straight line, but the blue light is gradually filtered out. Here is a schematic of that. You have all the wavelengths of the white light from the sun coming in, but then the blue light is scattered away, and you lose those short wavelengths, and by the time it reaches you, it is skewed towards the longer wavelengths, towards the red. The thicker that layer of atmosphere is, the more important this effect is. If you look away from the sun, it also causes the sky to appear blue because of this scattering. Yeah, so in the direction of the sun looks white, but outside of the direction of the sun, you see a blue sky. If the sun is setting or close to the horizon, then you see that the path of light that it needs to travel through the atmosphere is much longer, and by the time it reaches you, the light will be red, but only if you look in the direction of the sun. If you look away from the direction of the sun, the whole dome of the sky above you will be deep blue, and that is causing that increase in short wavelength if you look at the spectral composition of that Manuel Spitzchen showed. Here is another schematic that can make you understand this effect. It is very rare, because if people love these sunsets here with beautiful, red clouds, and it's actually very rare you see it like this. Either there's cloud cover all over, or there's no cloud cover. Sometimes there is scattered cloud cover, which allows the sun to penetrate its light below the clouds, and then it's reflecting from the bottom part, the downward facing side of the clouds. It's reflecting this red light, which is still in the spectrum, and the blue light is actually blocked by the clouds. So therefore, on these very rare occasions where you have partly clouded skies, you can see this beautiful red skies, but those occasions are actually very rare. The Mie scattering mentioned here is scattering by larger particles, and that is scattering in the direction of the light beam, and it's much more like a gray filter. It's not filtering specific shorter wavelength. And that Mie scattering is something that is happening by water vapor or clouds or larger particles. So Rayleigh scattering takes care of this blue enrichment during sunrise and sunset. Here we measured a similar thing with Tom Wouldes with a spherical spectrometer, photospectrometer on the roof of one of the buildings on the campus. And we also have time of day and spectral composition. These data can be used. Also the data in Manuel Spitzchen's paper can be used for analysis if you want. And during noon, we have a nice spectral composition of the sun. We measured these things with a spectrometer. We have a SpecBOS spectrometer, quite an expensive instrument with a fiber optic that we placed. We can place different light captors on. And in this case, we placed a spherical light captor on it, which is rather rare. Spectral compositions with a spherical light captor. It's important because then you do not measure the directionality of the light. Manuel Spitzchen used another type of captor, which is also less sensitive to directionality. And that is, I think, important because you don't want to make any assumptions about the direction that you look or so when you want to evaluate these light spectra. So that's the spectrometer. These are the data that Tom collected, and I show this graph because here it is important to see if you have wavelength here again. You can see here in the, let's look at the top row of the curves, if you distinguish between days when there is a clear sky in gray and days when there is clouds, you can actually see that the Mie scattering is actually causing a little bit more decrease in the lower wavelength. It's not it's almost a gray filter, but not exactly. There is a little bit more filtering of the longer wavelength due to Mie scattering, and this is causing a slight blue shift, as you can indicate here, as is indicated in the bottom row, a slight blue shift in the solar spectrum, especially around sunrise and sunset. Okay, so that's an additional effect. So what is important if you are evaluating light is, of course, we talked about wavelength already. Here you see the range of visible light. Light is an electromagnetic radiation, and it's part of a much larger spectrum that includes radio waves, if you listen to your old type radio, but also radar waves, infrared, of course, but things like ultraviolet and then X-rays or even gamma rays. They're all part of that electromagnetic spectrum. And the gamma rays, of course, are filled with a lot. Every photon in the gamma ray is filled with a lot of energy, whereas every photon in the radio waves is barely carrying much energy. So there is a relationship between the wavelength and the energy. And that relationship you can calculate this. It actually becomes important later on in the talk. So you can calculate this with the Planck's wave energy equation. I think, yeah. The units, the energy for one photon equals Planck's constant times the speed of light divided by the wavelength. And I make a little remark here. The speed of light is in meters per second. So wavelength should be expressed not in nanometers anymore, but in meters as well. And if you use this equation, you can calculate the energy per photon, which means that if you use a photometer, that typically measures not photons, but it measures energy. So watt per square meter. And using this equation, you can calculate how many photons are emitted or received by the object that you're interested in. And that relationship is plotted here. So a photon is nothing else than a wave or a particle that is carrying electromagnetic energy. All right. Where is this energy coming from? Well, it's coming from a sun, but also it comes from other stars, right? And those stars can be modeled, if you want, with what we call a black body light emitter. And here we see simulation and data for different black body emitters with increasing temperature. So those stars are pretty hot, 3,000, 5,000 or 10,000 Kelvin. And if that temperature increases, these are surface temperatures of stars, if it increases, then you see that the electromagnetic spectrum is actually shifted towards the shorter wavelength. So the more energy this light source has, the hotter it is, the more energy it has, the more energy-rich photons it will send out. By the way, this is wavelength in angstroms. One angstrom is 10. Do I say this correctly? Divided by 10, and you get nanometers. So here we see actually spectra from real stars, and you can see that some of these stars are so hot that they're really very, very strongly emitting ultraviolet light. Actually much more than our sun. So here you see also these typical absorption bands that astronomers are very interested in, because they say something about the atomic composition of the surface of the sun. So for instance, here you see the hydrogen bands, and that is something why astronomers are very interested in measuring these spectra. So very good spectra are emerging from astronomy. Here we have an interesting relationship. And this is, I think, the last slide I'm going to show you about stars. But here we see the relationship between the size of a star, and here we have the size expressed in sun masses. So there can be stars that are 30 or 60 times heavier than our sun. And here we see that it is, here are the smaller stars. And the larger the star is, the more UV light, the higher the temperature is, and the more UV light it will emit. Our sun sits somewhere here, and this means that the night sky, if our sun is, if we're in the shadow of the Earth and we don't see the radiation of our sun anymore, that the sky is actually dominated by the light from the sky is dominated by the larger stars emitting more ultraviolet light, and that is the reason why we get a relative enrichment during the night in the UV part of the spectrum. And that's nice to know, because that explains why flowers that are flowering during the night are good in reflecting UV light. So the colors that we don't see are actually observed by moths, by night active insects. And they have photoreceptors that are especially sensitive, that are very highly sensitive, full stop, but also very sensitive in the UV range of the spectrum, because that is a more dominant range. Okay, this is another table showing the similar thing I just said. The other light source we have at night is, of course, the moon. In full moon nights, we have the moon as a reflector, reflecting the rays of the sun. So sunlight is reflected from the surface of the moon. And here we have, in comparison, a sunlight spectrum, how it is perceived on Earth. So here, actually, I should go. Here is a ideal black body radiator with the same surface temperature as the sun. Here is what the sun actually emits before it enters our atmosphere, and this spectrum is the sunlight, how we perceive it on the face of the Earth. Now, if you compare that to the spectrum of the moon, and I try to get the axis, the wavelength axis in the same range here, if you compare it and you see that the moon is doing almost a perfect mirror of the sunlight, there is not much change in the spectral composition. So the moon mainly reflects sunlight without much filtering. Of course, that's because the moon has no atmosphere. So the additional nocturnal enrichment in the UV range really comes from the stars. The night sky with full moon, this is another fact, good to know. The night sky with full moon is at least 100 times brighter than with new moon, when there is no reflection coming from the moon. All right. And here are the data that show that. Here we have, this is again from Manuel Spitzen's paper, where he was able to analyze nights with different levels of illumination from the moon. And you can see that if you have full moon, we have this additional reflection of the sun in the spectrum. And if we have a new moon, we see this enrichment here, in the relative enrichment in the UV region with shorter wavelengths. And here the oxygen band again. And we have the infrared. If you do that in the city area, so that was the rural area, if you do it in the city area, you see that those spectra at night are looking very, very different, and they're really dominated by the city lights, by the stray light from the city, or the sky glow from the city, and you see that the moon is not changing that spectrum very much. So that indicates how important city illumination is for the spectral composition at night. You can actually easily imagine how a nocturnal butterfly that has to find its food source by this ultraviolet part of the spectrum, a moth at night has to find its food source by the ultraviolet part of the spectrum, how that moth is almost blinded by this huge light pollution from the cities. And that is, of course, something that is of interest for some of the BioClock projects. Here's another effect of artificial light at night. So here is a little image I put in. It's showing the sky glow, the reflection of the city lights against the bottom part, downward facing part of the clouds. Here we have a schematic. We have the city lights. In a cloudless sky, they would go straight up, but once you have clouds, it can be reflected down, and then you have extra disturbance from artificial light at night. Here we have measurements by Sjoerd van Hasselt. In our group, actually working, is now a postdoc with Peter Meerlo. And Sjoerd measured the spectra again using a spectrometer. Here you see the UV enrichment coming in, but you also see a lot of artificial light at night. And when you compare a clear sky in the black lines with a overcast sky, you can see that that relative intensity is enhanced, not in the UV, because that light is actually filtered out by the clouds, but the artificial light is reflected from the downward facing clouds back to the face of the Earth, and that is causing more disturbance. If you have a full moon, that effect is a little bit less, because of course the moon is the moonlight is penetrating through the clouds. The relative effect is less. Here we have the amount of overcast, and we have the light intensity that is coming back. So at night, the more clouds you have, if you're close to the city at night, the more clouds means more light. And the geese that he measured here, he measured sleep in geese. You can actually see that the non-REM sleep is disturbed, and they're sleeping less. They're spending less in non-REM sleep and with less power if there is more overcast. And we think that's an effect of this artificial light, because we don't see it when we have a full moon. And here's a schematic how we think that works. All right. I'm inclined to say, is this all clear, or are there questions by now? Because I'm entering a new part of the talk now. So if there are questions, maybe it is an interesting moment to ask some questions, if there are urgent questions. I see something in the chat. Welcome. Okay.
Yeah, no, the chat is right now clear of questions. So if somebody has one, just unmute. Else, Roelof, I think you can just continue and yeah, use the time.
All right, so that was part of light in nature. Of course, we do also experiments in the lab, and we're using many different light sources. And here I just, I wildly drew from the internet during this talk. So here we have a couple of light spectra. Here we have the sunlight spectrum that I already indicated. Here we have an incandescent lamp. So this is basically, you could say it's a little sun in a glass bulb, right? Because what we do there is we heat up a metal, and that the hotter the metal gets, the more blue enrichment you'll have, like the stars. And the colder the metal is, the more red enrichment you'll have. So typically those incandescent lamps are actually, they're showing a spectrum very similar to stars, and it will shift. You heat it up, it will be more and more white and more blue components. This is a relatively cold light source. So we called it a light source with a cold color temperature, probably, I don't know, 2000 Kelvin or something like that. And you can have light sources with 4000 or 5000 Kelvin, and they'll have more short wavelength light. But because you put in more energy, it's getting hotter, and the photons it's emitting is also carrying more energy per photon, so you get more shorter wavelengths. We have other types of light sources, so the high pressure sodium lamps. So here we see these big peaks here, those high pressure, and there are also low pressure sodium lamps that have other peaks, and actually those are the peaks that Manuel Spitzian saw in his spectrum with the city light pollution. These are typical old type street lamps that they're still using in the United States. And now they're getting more, in the Netherlands at least, more replaced by LED lamps that have a spectrum like much more similar like this. Do you see this little block? Is it in the way? Maybe. Yeah? So here we have a spectrum of a white LED, which is interesting. What makes an LED white? Well, first of all, the short wavelength LEDs are more efficient, electrically more efficient to produce photons. So the industry loves to produce a white LED by making a short wavelength blue LED and then putting phosphorus layers on top of it to create a phosphorizing effect that creates all these other shorter wavelengths. So typically in a white LED we have this blue component, which is important biologically because this blue component is right at the sensitivity, spectral sensitivity of our circadian photoreceptors. There are different types of other lamps, metal halides. There, of course, we construct lights also with different LEDs that can emit very narrow bands. And then we have the old fluorescent lamps with peaks here and some fluorescence here and phosphorus fluorescent here to make it a bit broader spectrum. But you can see these artificial lights generally can have different shape, different spectral compositions. And that poses a problem for us. So how do we measure this? Can we come up with a single measure that identifies the light intensity of all these light sources? Because we need to report about light intensity in our publications, because light is important for the circadian system. So it's very important for our field to know exactly how we report on these different light sources. And already now you see it can be pretty complicated. So the first lesson here that you could take away is measure the spectrum of your light source. Always measure the spectrum of your light source. And I think it should be reported in all the publications that we do, at least when light is important. All right. Let me see. Why am I not advancing? There we go. So, but now let's get into a part of the talk where I'm going to talk about how the circadian clock responds to light. And I'm going to build it up a little bit in a historical way, where I'm going to talk about the last 25 years or so. So in 2000, Aggelopoulos and Meisel published a paper that is hardly cited, but I think is very important at the time. And I don't want to go into too much detail, but what you see here is that the response curve of neurons in the SCN, these were in vivo measurements, the response curve of neurons in the SCN follows, is affected by whether you have a blue or red light adaptation. You see the relative response of SCN neurons in the red. This is if you have a blue light adaptation, you see that there's more response in the longer wavelength, and if you have a red light adaptation, you see that the response in the shorter wavelength is increasing. This at the time indicated already that the cones, the color vision system, so the cones are probably likely contributing as an input to the SCN. And here we have the curve at lower light intensities, where we see that there is a wonderful response that looks like rods. So Agalopoulos and Meisel in 2000 concluded cones and rods are the photoreceptors for the SCN. So what's the problem? You know, can we leave it at that? Well, there was a problem. And oh, by the way, this is another data that were collected. That was collected by Howard Cooper, where we see a large response in mice in the UV range, and so that indicated that the UV cone was projecting and involved in the circadian light responses. But what's now the problem? Well, the problem is that all kind of different mouse strains with retinal degeneration or rodless mice or coneless mice, so all the mice that have lost their classical photoreceptors, the rods and cones, they were still able to entrain to a light dark cycle, as you can see here. Here we have a C3H mouse line with retinal degeneration, and here we have a wild type, black 6 mice with normal retina, and we can see that both of these mice can entrain to a light dark cycle. So this indicated, this was done by A.B. Hauer in 1980, and this indicated that there must be another photoreceptor. I don't show the data, but it's also clear that if you remove the eyes, then there is no entrainment anymore. So it means that that unknown photoreceptor has to reside in the retina, in the eye. Here we have some data by Foster and Lucas around the same time, where they show that in these mice, this is a RDTACL coneless, so rodless coneless mouse, you see that the whole outer retina is gone. There are no photoreceptors, but you can see that this mouse has still normal circadian phase shifts. It has actually larger circadian phase shifts than the wild types. It has melatonin suppression that looks pretty normal, and it has a normal pupil response. So these are 3 non-image forming light responses that are actually not much affected by losing rods and cones. So there's a conflicting data set out there. So these are all the things I just said. Non-image forming light responses seem to be present in mice without rods and cones, and if you look carefully, it looks like it is a vitamin A based photopigment, so an opsin. It looks like an opsin with a peak absorption at 478. And then came Ignacio Provencio at the same time actually, and he isolated melanopsin from the melanopsin gene from the skin of Xenopus. Pretty smart move he made there. And with in situ hybridization, he could show that there's a melanopsin cells in the retina of mice, but also in humans. And you see here a cross section. It's a large cell type, a ganglion cell in a ganglion cell layer that spreads out through the inner and outer plexiform layers. So it receives on and off signals from the classical. It may receive on and off signals from the classical photoreceptors.
Roelof,
Yes.
sorry to interrupt. Maybe just 5 minutes left for your talk, so
Oh
you
no,
can wrap up.
Really? Oh, it's
so that
really?
we can give a chance to ask some questions also.
Yeah, where am I? Where am I?
But you can come again.
Ha ha ha. Well, let me fit. No, I think I can do this. So if you knock out melanopsin, you see that entrainment is still possible. I cannot go into details now. It's interesting, 2 independent studies. The one says melanopsin is not essential, and the other says melanopsin is critical, but the results are the same. If you knock out melanopsin, there is still entrainment. So Agalopoulos and Meisel are right, but also Foster and Lucas are right. It means that there is a melanopsin cell in the retina that receives input from the rods and cones and integrates it all, and that melanopsin cell is projecting to the SCN, and only that those melanopsin cells are projecting to the SCN. So that leaves us with a system, in humans at least, where we have 3 types of cones and we have rods, and we have the melanopsin cells. So we have 5 different photopigments that together create a signal for the biological clock. Now, how do we quantify light if we have 5 different photoreceptors? Especially if we do not know the precise wiring, and this is something we can talk on later, but that's important. So we don't know how these signals are interacting. And the solution that Rob Lucas came up with in 2013 and 2014 was, why don't we quantify each photoreceptor contribution separately? So we need 5 values to describe a light source. How do we get to these values? Actually, I really want to finish this part because it is getting important. So light measurements is very difficult. There's a lot of redundancy. There's a lot of units. It's very confusing, even for me when preparing this talk, but I try to find a path for you here. So first of all, we can talk about emitted light from a light source, and we can talk about incoming light to a surface or to the eye. Okay. So second of all, we can talk about the physics of light. So that's the energy or the number of photons. Or we can talk about the physiology of light. So that's the response of the eye. These are measurements in watts of photons, and these photometric measurements are measurements that relate to lumen or lux or candelas. If it's about the light source, we call it radiance. If it's energy, and we call it lumens if it's lux. If it's about the incoming light, we call it irradiance. And if it's about the incoming light relative to the human eye, we call it illuminance. So those terminologies should be absolutely clear. Once you know that, then you can understand the different measures. So here is the watts. All these are in red. The physics of light, energy is in watts. And because we know the relationship of energy and the number of photons, we can all calculate this using Planck's equation that I showed earlier into photons. If you want to talk about, so this is also watts of photons. If you want to talk about the response of the human eye, we should talk about lumen or lux. If you want to talk about the light source, you want that measurement to say something about the light source independent of the distance of the light source. So therefore, you're going to calculate it with a fixed, the amount of light that is either in the whole area that is emitted from the light source or on a fixed angle. And this fixed angle is called the steradian. Yeah, it's like a cone that is emitted. So it's in all the photons or all the light that is emitted from in this cone is calculated. So this area gets wider if you have a further distance from the light source. So these measurements, the emitted light here, the radiance and luminance measures are independent of the distance. But the irradiance and illuminance measures, they are dependent on the distance in a quadratic way. If the distance is doubled, the intensity per area is divided by 4. Okay, so intensity per steradian and irradiance and illuminance are intensities per square area. All right, so that's important. Now, how do you get, I have 3 more slides to go if you bear with me. Classically, how do you get from your spectral irradiance in watts per square spectral composition in watts per square meter? How do you get to your lux measures? Well, classically, we have a photopic response, our luminance efficiency function, and this indicates the sensitivity of the human eye to high light intensities. Scotopic if it's low light intensities. So what do you do if you want to calculate the amount of lux? You take your spectrum in watts, multiply it with your photopic efficiency function, luminous efficiency function, which are defined here by the CIE, and then you multiply it, and then the result will be this curve. These are the photons that are perceived by the human eye. And then you're going to integrate this curve and you get the total amount of lux after you have multiplied it with a constant here of 683. And what was the brilliant idea that, the problem, of course, this works only for the human visual system, not for other organisms, and also not for the non-visual responses like the circadian clock. So we had to come up with another measure, and the solution was to quantify each photoreceptor contribution separately. So we're looking to calculate 5 values. And this was the first approach by Rob Lucas. Here we have again our incandescent light. We have the 5 different alpha, we call it alpha-opic action spectra, absorption spectra, and that then, if you multiply them, that creates 5 different curves where photons are sort of perceived by the eye. And if you integrate those curves, then you get your alpha-opic measures. Then comes in Lux Slangen later on, who says, well, that's all fine, but we need to, in order to make this a CIE standard unit, we need to calculate this relative to a light standard, and this is a D65 light standard that is similar to the sun. And so what you do now is you're going to calculate the relative contribution of this curve relative to the standard. So you divide it by the standard, and that gives you 5 relative numbers. And here are those relative numbers. Here is basically it's the bottom line. Sorry, it's where am I? Yeah, here. It's the alpha-opic equivalent daylight illuminance relative to D65. That gives you these different numbers. And I think those are the numbers that need to be reported if you work with humans. Can you calculate this? Yes, very easily, because there's a great toolbox. I made this slide with the toolbox of Lux Slangen. Here I put in another light spectrum of a white LED, and you can see it is calculating these 5 curves, and then the relative contribution will give you these 5 numbers, and you can compare them, and it's an Excel sheet that you can use. It was very quick, but I hope it is clear. This is something that can be very helpful. This is the Light Measurement Handbook by Alex Ryer. You can download it from this website. And here is the toolkit I was talking about, developed by Lux Slangen and Rob Lucas and others. This is now the standard for light measurements, and you can use it fairly easy. You can put in your own measured light spectra, apart from standards that you can fill in. So you can also freely download it from this website. So my last slide, how do we measure light intensity in a way that is relevant for the circadian field? In my view, always measure the full spectrum between at least 360 and 780 nanometers with a spectrometer in watts per square meter per nanometer. Always publish the full spectral data in the log photons. So you have to measure it, but you have to publish it as well. And I like a representation with in a logarithmic fashion, because according to the Weber-Fechner law, this is how biological systems respond to light, either in watts or in photons. Now, for human studies, we can use this CIE toolbox and add those. So to the spectra, we can add the 5 alpha-opic values that I've introduced. And these are different terminologies for those 5 values for the different photoreceptors. But for animal studies, we have a problem. You can create your own alpha-opic toolbox if you have the spectral response curves of the photoreceptors, and you can add those species-specific alpha-opic illuminances by using something called the Govardowski absorption functions, and you also have to take into account the lens filtering characteristics then. Now, for rodents, this is not complete. For insects, we are still very far from having a standard for this. For rodents, we will actually talk in first week of February. We'll go to Manchester and try to develop a similar tool for rodents, which is now So that's under development, really. So there are people thinking about this at the moment. And that's it. I hope it was fast. It was a lot of ground to cover, but I hope I made some sense of it. And of course, the presentation will be available also, so you can look up some values on websites, etc. Thank you.
Thank you very much, Roelof, for this very interesting talk. It's clear that there is a lot to say about it, so we will have to invite you another time, I guess.
Well, of course, Lux Langen is also somebody who knows a lot about it.
Yes, perfect.